So today we are going to discuss about cereal and cereal products, their individual constituents, including proteins, lipids, carbohydrates, and vitamins in cereal flour and their relationship in dough making. You know, cereals are staple foods in our daily diet that not only satisfy our hunger, but also provides all the required nutrients for essential for growth, maintenance, and growth development and maintenance for our body. Generally, cereals belong to species of wild grasses. Major cereals include wheat, rye, rice, barley, millet, and oats. Wheat and rye have a special role since only they are suitable for bread making. In industrial countries, mostly people consume bread, which is ready to eat foods and doesn't require much time and energy to prepare. And that bread provides 50% of the daily requirement of carbohydrates, one third of the proteins, and 50 to 60% of vitamin B complex, minerals, and trace elements, which is required for our body. Yet you can see varieties of products, baked products prepared from wheat. Now let us see the composition of wheat grain. Basically, there are three parts, that is bran layer, endosperm, and wheat germ. Bran layer contains almost 7 layers, including epidermis, hypodermis, cross cells, tube cells, seed coat, nuclear tissue, and aileron cell that is just touching the endosperm layer. And these bran layers are partly waterproof, which ensure that the seed will not start to grow again immediately after harvest. At one end of the grain is the wheat germ. That is the young plant that will grow when the grain is put into the soil and given the right conditions of moisture and warmth. The aim of the miller in making white flour is to obtain the greatest possible amount of the whitish interior of the grain, that is endosperm part basically. This is endosperm. Maximum part covered in the grain is endosperm, responsible for providing white flour. And bran is not, in fact, wanted mainly because of its color and texture by the food industries, whereas germ is not wanted because of its high fat content, which reduces the keeping quality of the baked food product because of development of rancidity during storage. In structure of cereal grains, embryo is the small part and attached to the base of the seed you have seen in the diagram. Root and leaf of new plants are formed from this embryo only, and this part is rich in lipids and high in total nitrogen and ash content. Next and most important part is endosperm, which makes up the major portion of the seed, and it supplies the sprouting embryo with food in the period before the root and leaf begin to function. Endosperm cells consist mainly of starch and proteins. The starch is form of spherical granules embedded in a matrix of protein, and this endosperm is main source of white flour as well as semolina. These are products of wheat. Next part is bran, which is covering the whole wheat grain. This is dark colored and consists of several layers, that is 7 layers, as you have already seen, and it consists mainly of dietary fiber. Layer of cells surrounding the endosperm is known as aleurone layer. In milling, the aleurone layer separates with all the seven layers of the bran. Bran has unusually high percent of crude fiber and ash. Crude fiber includes all the dietary fiber parts, that is cellulose, hemicellulose, as well as lignins present in the wheat grain. Composition of cereal grains varies depending on their variety, geographical situation, and other conditions. Major compositional features. Major composition of wheat naturally include macronutrients as carbohydrates, proteins, lipid, as well as various micronutrients, that is vitamin B complex and various minerals. 80% of the dry matter of cereals is composed of carbohydrates, that too mainly of starch, and some amount of dietary fibers present in bran layer specifically. Wheat grains contain 10 to 12% of protein, which is found in all tissues of the cereal. Main problem with cereal grains is it is having lysine as limiting amino acid. Lysine is essential amino acid, but its level is very low in almost all cereals, whereas methionine, this is also essential amino acid, and its content is low, particularly in wheat, rice, barley, and corn. Lipid content is also very low in most of the cereals. In wheat and rice, its amount is very low, that is ranging from 1 to 2%, whereas maize contains almost about 3%, and oat contains 5.7% of the lipids. More of the lipids are present in germ layer and some in the bran of the grain. Out of different lipids, linoleic acid, which is essential fatty acid, is predominant fatty acid in cereal lipids. Regarding minerals, about 90% of the minerals are present in the form of phosphates and sulfates of potassium and magnesium. And important point about cereal grains is that they are poor sources of most required minerals, that is calcium and iron. Calcium naturally required for skeletal tissues, for bones, etc. And iron is very important part of hemoglobin. But cereals are poor sources of both of these minerals. But whole grain cereals are important sources of B vitamins, which is mostly present in bran layers. So refining or polishing of the grain reduces its content. Cereal grains contains various enzymes also, including amylases, proteases, lipases, and oxidoreductase enzymes. All of them play important role during processing. Now, we will discuss about individual constituents of wheat and their importance in baking. First of all, let us talk about proteins. T.B. Osborne was the scientist who separated wheat proteins in 1907 based on their solubility into 4 fractions, that is albumin, globulin, prolamins, and glutelins. Albumin portion of wheat is known as leucocin, and it consists of 14.7% of the total wheat protein, whereas globulin portion known as adenosine consists of only 7% of the total wheat protein. Prolamins is known as gliadin in wheat, which consists of 32.6%, and glutelins, which is known as glutenin in wheat, it consists of 45.7%, and both these gliadin and glutenin plays important role in baking. We will talk in detail. Wheat flour contains soluble and insoluble protein fractions. Soluble proteins which comprises only 20% of the total proteins, include obviously albumin and globulins and certain minor glycoproteins. These proteins do not contribute to dough forming properties of the wheat flour. Insoluble wheat proteins, that is prolamins and glutelins, exist in the ratio of 2:3, and they are also referred to as gluten proteins and play major role in dough formation and baked products. These two, prolamins and glutelins, are major storage protein in wheat. It is heterogeneous mixture of gliadin and glutenin. Gliadin, that is prolamin, and glutenin, that is glutelins. Gluten is formed when water and flour mixture is needed to form dough. Gluten proteins are responsible for formation of viscoelastic dough that is capable of entrapping gas during fermentation, and because of this entrapped gas during baking, it will leave the material, and final baked product will be spongy and its volume will also be increased. Both fractions of insoluble wheat protein in hydrated forms have different effect on rheological characteristics of the dough. Prolamins are responsible for viscosity, whereas glutelins are responsible for elasticity of the dough. Gluten proteins in association with lipids are responsible for cohesive and viscoelastic flour properties of dough, which is suitable for making bread and other bakery products. Gluten consists of mainly protein, that is 90%, and 8% of lipids and only 2% of carbohydrates. Now let us see mechanism of dough and gluten formation. Several physical and chemical transformations take place during mixing and kneading of the mixture of wheat flour and water. Under the applied shear and tensile forces, gluten proteins absorb water and partially unfold in order to facilitate hydrophobic interactions and sulfhydryl disulfide interchange reactions that results in the formation of thread-like polymers. And these linear polymers in twin interact with each other, presumably via hydrogen bonding, hydrophobic association, and disulfide links in order to form a sheet-like film which is capable of entrapping gas. Therefore, optimum ratio of prolamins and glutelins are always essential to form a viscoelastic dough. Now comes lipids. You know, cereals contain very low amount of lipid comparatively, which is present in germ and bran mainly. In wheat kernels, germ and aleurone cells are rich in triglyceride portion of lipids, whereas endosperm contains maximum amount of phospholipids and glycolipids. On an average, the wheat germ contains 6 to 11% of the lipids, total lipids present in wheat, whereas bran contains 3 to 5% of the lipids, and endosperm contains very, very minute amount, that is ranging from 0.8 to 1.5% of the total lipid of the wheat grain. Wheat flour contains 1.5 to 2.5% lipids depending on the milling extraction rate. Flour lipids are differentiated by their solubility. Non-starch bound lipids consists of 75% of the total lipids of the flour, whereas starch-bound lipids consist of only 25% of the total lipids present in the grain. Non-starch and starch-bound lipids differ in their composition in the way that non-starch-bound lipids consist of triglycerides and glycolipids portion, whereas starch-bound lipids consist of lysophosphatides. Non-starch lipids are further fractionated into free and bound form by solvent extraction. Both free and bound lipids contain non-polar glycolipids and phospholipids. Free lipid fraction contains 90% of the total non-polar lipids and 20% of total polar lipids of the wheat flour. By kneading the flour into dough, glycolipids becomes completely bound with gluten, while binding of other lipids is only 70 to 80%, resulting in a starch protein lipid complex matrix. Non-starch lipids affect rheological properties of the dough, whereas starch-bound lipids affect properties of the baked products. Lipids are enclosed within the amylose helices, where lipids complexed within the starch granules retard swelling and increase their gelatinization temperatures. Thus, they influence the baking behavior of the cereal and properties of the baked products. Polar lipids positively influence the gas holding capacity of the dough and baking volume, while non-polar lipids generally negatively influence the baking results. Next comes carbohydrates. Its major portion is covered by starch, which is major storage form of carbohydrate and exclusively occurs in endosperm cells. It consists of 25% amylose and 75% of amylopectin. Starch granules swell when heated in water suspension. At the end of swelling, they lose their native structural form and arrangement. That is, they are gelatinized. Starch contributes viscoelastic strength of the semi-rigid structure formed along with gluten during baking, while lipids and proteins are heterogeneous constituents of starch granules. It contains other polysaccharides also in the form of hemicellulose, pentogens, cellulose, beta-glucan, and glucofructans, which is present primarily in the constituent of cell wall and are more abundant in the outer portion of the seed kernel. Next carbohydrate portion is in the form of pentogens. Its content in wheat flour is 2 to 3%. A portion of pentogens is water soluble and absorb 25 times more water and form highly viscous solution. Insoluble form of pentogens swells extensively in water and increases crumb juiciness and chewability of the baked products. Pentogens play an important role in baking quality since they also participate in the gluten formation. Next is beta-glucan, and its level is also very low in wheat flour, that is ranging from 0.5 to 2%. Beta-glucan is linear polysaccharides with D-glucopyranose units linked by beta-1,3 and beta-1,4 linkages. These are polysaccharides also known as lignans. They are slightly mucus in nature and provide high viscosity to the water solution. Next comes sugars, which includes mono, di, and trisaccharides, as well as other low molecular weight degradation products of starch. Sugars occur in relatively low concentrations, but are important for dough leavening. character in the presence of yeasts. Cereal grains also contain a number of enzymes, including amylases, proteinases, lipases, and oxidoreductases. All of them play important role in cereal during processing. Amylases, specifically alpha and beta amylases, are present in all the cereals, and their optimum activities are desirable in dough making in presence of yeast. Acid proteinases are present in wheat flour, and it is involved in cleavage of gluten bond, thereby affecting the softening or mellowing of gluten during baking process. Next comes lipases, which are responsible for the fatty acids appearing during the storage of the cereals and their products. Here in this table, you can see chemical composition of cereals and cereal products. Here you can see that moisture content ranges from 11.2% in Italian millet to the maximum level of 39%. That is in wheat bread, be it white or brown, both are having same amount of moisture. Whereas protein content ranges from 6.4 in rice parboiled milled to 12.3% in the Italian millet. Lipid content ranges from 0.4 in rice parboiled milled rice to 5% in bajra. Carbohydrate content ranges from 49% in brown wheat bread to 79% in milled parboiled rice. That is maximum amount. Whereas crude fiber level ranges from 0.2% in milled raw rice as well as milled parboiled rice to 3.6% in ragi. Ragi is very rich source of crude fiber. Similarly, mineral content ranges from 0.6% in milled raw rice to 3.3% in Italian millet. That is all about chemical composition range in different cereals as well as cereal products. That's all. Thank you. Thank you very much.